Now that it's possible to derive stem cells from patients, we can take those stem cells and turn them into a lot of different cell types in the body, such as heart cells, which is the main focus of my lab. So this is really powerful because now we can study how a certain drug affects a particular patient, or be able to study a disease by studying cells from a patient with that disease. The heart is very soft and flexible, right? As the heart cells contract, they can actually pump the blood, and the cells are aligned into a muscle tissue. So we want to be able to capture those features of the environment that the cells experience in the body in the dish. And so we use a lot of different biomaterials and tissue engineering techniques to be able to model certain physical features of the human body within the lab. So what we do in my lab is fabricate these silicone wafer templates. So these have ridges in them that are about the width of a single cell. So from this wafer template, we can cast rubber stamps and then use those rubber stamps to pattern proteins onto the substrate. Then when we add our heart cells on top of that substrate, they stick only to where we printed those proteins. So we're able to dictate the architecture of cells and tissues in the dish. So that's really powerful for being able to mimic the architecture of cells and tissues in the body in the lab. Another key aspect of what we do is to be able to measure the function of these patient-derived stem cells and their derivatives. So the cells in the heart, for example, are contracting and pumping blood. And so ultimately we'd like to see if a drug or if a different genetic mutation affects the ability of a heart cell to pump. So if we focus just on if the heart cell is alive or dead, that doesn't really give us enough information. So we use a lot of different engineering approaches to be able to measure the contractile activity of heart cells and be able to compare that based on different patient genetics or different drug treatments. By combining patient-derived stem cells with our engineered platforms, where we can collect a lot of physiological data in a very quantitative, reproducible way, we can start to develop drugs that will work for particular patients or particular groups of patients based on their particular genetic background, be that what type of disease they have, or even things like gender.